Okay, this is Lab 8, the digestive system. And this is Digestive System 2 because we're going to look at more of the chemistry, the enzymes from each organ that are utilized for digestion. All right, as well as a few other exercises in addition to that. Okay, so let me see if I can get this share to open up now. Here. Yeah. This way. All right. So here we are in Lab 8, Digestive System Part 2. Right. Exercise 1 are the physical mechanisms of digestion. And so we have these terms, physical mechanisms, for peristalsis, segmentation, gastric churning, haustral churning, degglutination, and mastication. This really comes from the textbook, and it has, a certain amount of, I guess, clinical importance to it. You are to identify, whether it functions mostly as digestion or indigestion or propulsion, pushing something forward. And so we're either going to use a D or a P for the description, and then the organs associated with it. So peristalsis, that would be P for propulsion. And the organs include the pharynx, or actually, I take that back. It really should just be the proximal end of the esophagus through the rectum. Segmentation is D for digestion. That occurs in the small intestine. Gastric churning, D for digest, stomach. Haustral churning, D for digests, large intestine. Degglutination, P for propulsion, in the oral cavity of the mouth. And the pharynx, so this is swallowing. It starts in the oral cavity and continues through the pharynx. And then we have mastication, which is chewing. That would be D for digest. And in the oral cavity, teeth. I have to admit that if I asked a question, I think the only time I've ever really asked about the type of mechanism, physical mechanism of digestion occurs here, it's generally either the small intestine for segmentation, gastric churning for the gastric, the stomach, and haustral churning for the large intestine. So if I pointed to the stomach and I asked you what kind of physical mechanism or type of digestion, physical mechanism, let's say, of digestion occurs at the stomach, right? We're talking about gastric churning. If I ask the same question from the small intestine, let's talk about segmentation, the large intestine, haustral churning. Okay, then we go on to number 2. These are the hormones. So when we were looking at the images in Lab 7, and when it came to the stomach and the small intestine, some of the cards were referring to some of the hormones at that point. Even though it wasn't part of the content of that lab, it was kind of included in that lab. And I said that we would come back to that because those are topics that are covered in the next lab. The same way, I believe, when we're looking at the stomach, it mentions about gastric churning, the small intestine, segmentation, the large intestine, the haustral churning. But it really, again, is part of this lab, not really last week's, but images in this week's, that we're going to look at, hopefully will give us that same reference. So you're tying together the contents of the lab we're filling out now with the images on the course page. Okay, so we have 3 hormones, and we're not limited to those. There are other hormones, but we're going to look at 3 hormones: gastrin, CCK, or cholecystine kinase, and secretin. So we'll start with gastrin. The production site over to the left-hand side are the glands in the stomach mucosa. It releases the hormone gastrin. The target site are glands in the stomach mucosa, because it's produced in the stomach, and its target are other cells in the stomach. And the action would be increased secretion of gastric juices, increased stomach motility. So the actions, those three phrases there, I guess I could have put maybe an asterisk in front of them. Those are the actions that you're going to put into one of the empty squares here to the far right of each of the hormones. So gastrin, right, the action is increased secretions of gastric juices, increased stomach motility, which is your second option here. Okay, then we have cholecystine kinase is produced in the small intestine. It targets the pancreas, the gallbladder, and the gallbladder was kind of important, the hepatopancreatic ampulla and sphincter. Arguably, both CCK and secretin target those two things. The action is to stimulate the release of bile and pancreatic juices and the gallbladder. And so if it's targeting gallbladder, we want it to stimulate the gallbladder to do something, and opens up the hepatopancreatic sphincter of Oddi. So what it's not telling you here is really why it's being released in the first place. And CCK is mostly responding to the presence of fats and proteins in the chyme that's being released from the stomach and entering in the small intestine. So CCK is trying to make sure you get enough bile from the gallbladder and digestive enzymes from the pancreas to be passing through the sphincter of Oddi, or the hepatopancreatic sphincter, because there are now clearly substrates there that need to be digested. Secretin, the stimulus, is a little bit different. It primarily responds to the pH value of the chyme that's entering the small intestine. And so when the pH is dropping, because you now have chyme entering there, and of course the chyme is full of a lot of hydrochloric acid, that one of the things you want to do is very quickly neutralize that chyme in the small intestine for multiple reasons. And one is you don't want it to start burning holes through the walls of the small intestine. We have mucus secreted along the entire digestive system, but everything, any area can be kind of be overwhelmed. So you really want to alter the pH to keep from eroding, the walls, the mucosal walls of the small intestine. But you also want to elevate the temperature elevate the pH, because as the enzymes that are stored in the pancreas, are delivered into the small intestine, you don't want to denature those proteins. Remember, enzymes are proteins, and proteins are vulnerable to what? High temperature, low pHs. And so if it's too low a pH, then you denature, the hydrogen bonds of those proteins, and now they're no longer functional. So it's actually for multiple reasons. All right, so we're looking at the production site, which are, again, small intestine glands found in the small intestine, similar areas, CCK. The hormone is secretin. It targets the pancreas and the gallbladder and the stomach. And that's where, like, the stomach is different than, like, including gallbladder for CCK up here. So that function is the first one, or the action, I should say, is the first one. Stimulates secretion of bicarbonate from the pancreas and the gallbladder. Other enzymes are going to come with it because they're all mixed, and are present in the pancreatic duct. But its primary focus is to get the bicarbonate in there to elevate the pH. It also feeds to the stomach, and essentially inhibits gastrin from being liberated in the stomach, because you already have enough acidic solution coming into the small intestine. So, it not only stimulates the pancreas to start bringing down some bicarbonate to neutralize that hydrochloric acid, or the lower pH of the chyme that contains a lot of hydrochloric acid, but it also feeds on the stomach to inhibit, gastrin secretion for a while. Okay, then we move on to digestive aids. That too was included in, some of the flashcards, from the notes last lab. So we're going to look at these digestive aids and we're going to identify the production site and basic actions. Right, so bile. Bile is produced in the liver, and you can say emulsifies fats, emulsification of fats, or emulsify fats. Hydrochloric acid, produced in the stomach. Now, hydrochloric acid serves a few different functions. Again, because of that low pH, it helps start to break down proteins, by breaking up the hydrogen bonds. It stimulates the conversion of pepsinogen to pepsin. That's where we begin protein digestion. It also, remember, helps kill bacteria, and microbes that might be susceptible to low pH. So it has a couple different functions. Bicarbonate, produced by the pancreas, neutralizes the chyme. Mucus, production site. This is along the entire digestive system. The mucosa along the entire digestive system releases a lot of mucus, and for multiple reasons. Kind of lubes the tube, so food can pass through there. But remember, mucus is a very good physical barrier, or acts as a physical barrier for microbes. Then saliva, the production site are the salivary glands, parotid glands, sublingual, submandibular glands. And the action is to moisturize food, but also it begins carbohydrate digestion in the mouth. Okay, so now we move on to the little more challenging aspect and how you're going to use it. I think it'll be, better illustrated when we look at some of the pictures. But here we're going to identify the enzyme and the source of the enzyme that metabolizes these different molecules. So we're looking at the carbohydrate family, the lipids, the proteins, and nucleic acids, just like we did in the chemistry chapter in A&P1. And, in there we identified what are the building blocks of carbohydrates, what are the intermediate products, what are the macromolecules. We looked at the bond types. We looked at, basic functions of those things. We did that for, and so in doing, we identified the monosaccharides, the disaccharides. Then you moved on to proteins, and we looked at the building blocks: amino acid, dipeptides, polypeptides, basic functions. Well, you come back to that again, and I have a chart down on the bottom, and you essentially are doing the same thing. We're not worried about the bond types, and we're not really worried about functions. What we're doing, and we're adding to this chart, is identifying the organs that produce the enzymes, or enzyme, or organ, maybe only one, that takes maybe the macromolecule to an intermediate product, and an intermediate product to a building block, in the case of a carbohydrate and protein. Remember, lipids and nucleic acids, they just go from a macromolecule to a building block. So it'll be, one enzyme and one source for that enzyme. Okay, so here we're going to start by, we're going to look at carbohydrates. Right? The enzymes that we're going to put over the arrows, because, we're going to write kind of chemical reactions, will be amylase. We have two types of amylases, right? We have salivary amylase, and we have pancreatic amylase. Amylase that's released into the mouth, amylase that's produced by the pancreas and liberated into the small intestine. We have maltase, lactase, and sucrase. Our production sites may be, or are for these, the small intestine, the mouth, and the pancreas. And so these are the things we're going to insert in arrows and things like that. When it comes to the lipids, the enzyme will be lipase, and the production site is going to be the pancreas. When it comes to proteins, we're going to do this in kind of a two-step process. Breaking down proteins is actually kind of 4, if not more steps. It's really quite complicated, but we're going to kind of treat it like we do the carbohydrates, where we start with a macromolecule to an intermediate product to a building block. Because remember, we need to know how we break these molecules down to building blocks, because it's the building blocks that can be absorbed in the small intestine. If we don't break these molecules down to digestible, absorbable sizes of these molecules, then they're never going to get into the bloodstream. Okay, so our nucleic acids, our enzymes are ribonuclease or RNase, and deoxyribonuclease, kind of hard to say, DNase. And the production site is the pancreas. Okay, so here we have vertically, I have now, there are some blanks you're going to fill in, and you're going to use these enzymes and production sites that I just listed for carbohydrates, lipids, proteins, and nucleic acids in these blanks. All right, so if we look at the first family, which is the carbohydrates, right? We have, going across the top legend there, so to speak, we have carbohydrates, proteins, lipids, and nucleic acids. Over to the left side, vertically, we have the building blocks, intermediate molecules, and macromolecules, okay? So, if we look at the carbohydrate family, and let's just kind of stay in that one row, I've kind of color-coded some of these things as well for you. So if we start at the bottom, because digestion starts with the macromolecules and breaks down into intermediate, if not building blocks right away. And the arrows point to the direction in which we're moving, from a macromolecule to an intermediate molecule, that blue arrow there, and then from the intermediate molecule to your building blocks. Remember, arrows point to the products, so they start by the reactants, and they end up pointing to your product. So we're starting with big things, we're ending with smaller and smaller things. All right. So what goes into this line just above starch? That would be polysaccharide. So we start with a polysaccharide in the carbohydrate family, example of which is going to be starch. We have 2 enzymes that break down that polysaccharide to the intermediate product, which is the disaccharide. And the disaccharides are listed below: sucrose, maltose, lactose. Okay, so we have 2 enzymes that perform that same process. We have salivary amylase from the mouth, that's why it's in red, and we have pancreatic amylase in kind of a pink color. Okay, so we have either an enzyme from the mouth or the enzyme from the pancreas that's going to break down that polysaccharide, like a starch, to an intermediate molecule called a disaccharide. Technically, starches will break down just to maltose. Right? Sucrose is an example of sugars in some fruits and things, and then table sugar. Lactose is the sugar found in milk, lactose. So those are disaccharides. But now we need to break them down, into their building blocks, right? So the blue arrow points to the building blocks. So in that arrow, we write in monosaccharide. And your monosaccharides are glucose, fructose, galactose. Glucose, fructose, galactose. So how do we get from sucrose, maltose, or lactose to those? Well, we have enzymes in purple here: sucrase, maltase, lactase, and they come from what's called the brush border cells in the small intestine. So after you pass the pancreas, you're going to keep moving down the small intestine, and you're going to enter an area where we have cells that produce sucrase. The ending ASE tells you it's an enzyme, and the first part, SUCR, is indicating its substrate. So sucrase breaks down sucrose into its building blocks, and the building blocks will be glucose and fructose. Right? Maltase breaks down maltose to 2 glucose molecules. And then lactase acts on lactose to break it down to its 2 monosaccharides, and that is glucose and galactose. So the carbohydrate family and the protein family, I'll just tell you, is a little complicated. And then the last two really roll. All right, so I had color coded, as by the time we get to the end of this, you'll see how these enzymes are color coded by their source. The one from the, there's only one from the stomach, and that's in red. We have in pink here, pancreas, pink for pancreas. The purple here, sucrase, maltase, lactase, are coming from the small intestine, and so does dipeptidase over here in the protein family. And then pepsin, the only one in green, is from the stomach. So, this is kind of handy to repeat over and over again or draw out. And if you color code them with either, colored pencils or felt tip pens of some kind, you start really grinding in, where these, you can just quickly label up and create this, this chart. And by color coding it, you know what organ produces it. And you can create whatever color code you want. I would just have one for the small intestine, one for the mouth, one for the stomach, and one for the pancreas, and you'll be fine. Or you can write those terms in there, like next to salivary amylase, just put in parentheses the mouth, pancreatic amylase, the pancreas, so forth and so on. Okay, so let's go to proteins. We start with the macromolecule of a polypeptide. An example of that is albumin. We're going to break this down. Now, it begins in the stomach with pepsin. So as long as that food is churning around in there, remember we secrete hydrochloric acid, and that converts pepsinogen to pepsin. And pepsin starts to break down those long protein chains down to dipeptides. And once, so not all of that protein will necessarily be broken down, because proteins are rather complicated and they're very large molecules and long strands of things, sometimes 100, 200 amino acids. So that, it takes a while to break them down into 2 amino acid segments. Just think of that mathematically. So we might be, those proteins maybe are in decline and entering the small intestine before this digestion has been completed, changing it from a polypeptide to a dipeptide. So from the pancreas, what's found in the pancreatic secretions there, or the pancreatic juice, is trypsin and chymotrypsin. And so that will help complete breaking down that long polypeptide chain to dipeptides. Now, also from the pancreas is dipeptidase, and that starts acting upon dipeptides to break them down into amino acids. That's from the small intestine. And then the carboxypeptidase from the pancreas breaks the dipeptides down, or actually, I'll take that back. It cleaves off what's called the carboxyl group, at the end of this amino acid chain. So let me just repeat this, because I don't know if I just said that correctly about the source of dipeptidase. And if I didn't say that correctly, I apologize. But let me just clarify. We have these dipeptides, and we have longer proteins, that are entering into the small intestine, that are passing the pancreas in the small intestine. They're being acted upon by an enzyme from the stomach and 2 enzymes from the pancreas to help break it down to a dipeptide. And then from there, the dipeptides are broken down by dipeptidase, which is from the small intestine, and carboxypeptidase, that is from the pancreas. And the carboxypeptidase essentially cleaves off what's called the carboxyl group, the COOH group. The dipeptidase breaks down the dipeptide to amino acids. Okay, then we enter the lipid family. This becomes much easier. We start with triglycerides, and we end with the building blocks here. So the building blocks are 1 glycerol and 3 fatty acids. 1 glycerol, 3 fatty acids. Those are our building blocks. All right, so what enzyme facilitates that, or enzymatically pushes that reaction? Lipase from the pancreas. And then in the family of nucleic acids, we have DNA and RNA, which are the macromolecules. And from the pancreas in pink, we have DNAs and RNAs that break them down to their building blocks of nucleotides. Okay, so again, these are the enzymes. The organ that produces them is by color coding. And we're going to put them, so you can put those into any sequence you kind of want, the way, if you want to create another type of, well, let's say diagram that takes the carbohydrates only and focuses on those. And then maybe, again, because it's color coded, maybe draw like a small intestine, put a model of a small intestine next to sucrase, maltase, lactase, so you know where it's coming from. Maybe, a pancreas next to the pancreatic amylase or trypsin, chymotrypsin. But I think if you just, this is the way I teach this, without the enzymes, of course, in A&P1, when we talk about chemistry, we have it similarly set up here where we fill in all this and we identify on the bottom primary bond types and these functions of these different molecules. Now we're doing the same thing, but just entering the enzymes that break down these molecules. Okay, so hopefully that was a little more clear. Let me just see. Okay. I'm just looking at something. Okay. So let me stop this share, and then I'm going to reshare. Just a little bit smaller. And go into the course page, and we are going to it's not getting this right. Hold on. Let me just squish this. We're going to go look at the pictures, the images. Let me just get into my course content and get into the right page. There we go. Much better. Click on the first image so it'll be easier. There we go. All right. Okay, so these are from the laptops, the lab tabletops in the lab room, where we set up models to represent these different molecules that we're going to be looking at, these macromolecule, intermediate, and building blocks of the different families. And remember, in A&P1, you looked at models of these, the ball and stick figures, as well as maybe bottles of let's say sucrose, with a label on it, that we use those for examples of these disaccharides or monosaccharides or dipeptides, let's say. So those come back, those ball and stick models come back. So generally what we would have is a carbohydrate region where we'd start with the card on the left that says a starch or polysaccharide. Glycogen should be kind of scratched out on there because this is not how we break down glycogen. We're really just looking at the polysaccharide or starch. So above this, and perhaps on a subsequent picture here, you might have a jar, or a box of starch, that you would cook with. Or maybe, what are those ball and stick organic molecules of polysaccharide? And it represents the macromolecule form. So then from there to the disaccharide or a disaccharide form, which is over on the right-hand side, we have the arrow that represents the hydrolytic chemical reaction that's occurring, the enzymes, and the source of the enzymes. So we have over that arrow, the enzyme is from the salivary gland, that would be salivary amylase, and the enzyme from the pancreas, that would be pancreatic amylase. Okay, so salivary amylase, pancreatic amylase. Now we go from the disaccharides, which are the molecules on the left-hand side, maltose, a disaccharide, sucrose, a disaccharide, lactose, a disaccharide. And now we're going to break those down to our building blocks. So the little card says, what enzyme from the small intestine, is going to act on maltose, sucrose, and lactose. And I'll just tell you right now, it's telling you also that maltose breaks down to, so if you go from the left side, which says maltose card, and you go to the right side, it breaks down into 2 monosaccharides, 2 glucose molecules. Sucrose, when sucrase acts on it, breaks it down into glucose and fructose. And then lactose on the bottom breaks down into glucose and lactose. Okay, so we have maltase, sucrase, lactase, all from the small intestine. So an example of a question might be, there's a bottle, maybe there's be like a little bottle, with a label on it that says sucrose. Right? So you go, oh, so the question might be, is that a polysaccharide, a disaccharide, or a monosaccharide, or a macromolecule, an intermediate molecule, or a building block? Well, it would be an intermediate, or be a disaccharide. What enzyme digests that molecule, which means what enzyme is going to target and act on, use sucrose as its substrate? Well, that would be sucrase. What is the source of the enzyme that acts on, or utilizes sucrose as a substrate, or a reactant? That would be sucrase. What organ produces it? Small intestine. What are the products of digestion of a sucrose molecule? Glucose and fructose. They're kind of different ways to say the same thing. But if you practice writing this out, it'll make a lot more sense, because all we're doing is we're asking you, what's the enzyme that acts on maltose? Well, it'll be maltase. What organ produces that enzyme? What's the end product of the digestion? And of course, digestion means breaking all these things down. The digestion of maltose, it would be 2 glucose molecules. Then we're in the protein family. We have polypeptide. An example is albumin. So it's asking, what enzyme from the stomach will break down one of these polypeptides to a dipeptide? And maybe what enzymes, because there is more than one, from the pancreas. Right, so here we have one of those ball and stick models. Not sure why he didn't include the ball and stick models in the carbohydrates, because they're really easy to see too. But in my notes, in the PowerPoints, it includes some of those. So we're going to zippy through that like we did the last time. I won't belabor a lot of it, but I'll show you those images. So when you can see more than one, and here we have 2 blue balls here, which represents the amine group of an amino acid. These are 2 amino acids, which means it's a dipeptide. It would have been nice to have a polypeptide included here, in the polypeptide region. That generally is a 3 blue ball chain. There would be 3 amino acids in these ball and stick or straw models that we have here. All right. So we have from the stomach, we have pepsin, and from the pancreas, we have trypsin and chymotrypsin. We start with a polypeptide, we end with a dipeptide. Right, then we start with the dipeptide. So here we have a better view of this molecule, where you have this double bond here at that carbon, to close to the top of the model. And then you have a nitrogen blue ball, another double bond, and then a nitrogen blue ball at the end. So that is 2 amino acids, dipeptide. And then over to the right, we have 1 amino acid. We have 1 blue ball. We see that double bond there at that carbon. We know it's 1 amino acid. So it's asking, over the arrow, the enzyme from the small intestine that will be used to help break down the dipeptide to the amino acid, and what enzyme from the pancreas. So they're going to be 2. And we have dipeptidase, and we have carboxypeptidase. Dipeptidase is from the small intestine. Carboxypeptidase is from the pancreas. Okay. Here, now, or I should say now, we're in the lipid family. So to the left-hand side, we have triglycerides. It has a little container on the label. It shows you a bottle of Crisco. So that represents fats. Right? That's a triglyceride. Here above that, you can see a ball and stick model. Right? It's a rather large model. Kind of looks like a tripod or an octopus. Well, I guess it wouldn't be an octopus because it doesn't have 8 legs, but it kind of looks like a tripod. And that is a single kind of enzymatic reaction. Once we emulsify it with bile and we break it up into small little globules, then lipase from the pancreas will break it down into its building blocks. 1 glycerol molecule, and so that's your ball and stick model of a glycerol, the 3 carbon chain like that, in linear 3 carbon chain. And then we have the fatty acids, which are a longer chain, linear chain, with a double bond here at that carbon. 1 glycerol, 3 fatty acids. Okay, so the enzyme is lipase from the pancreas. The last family are the nucleic acids. The macromolecules to the left-hand side are DNA and RNA. And I feel certain that you pretty much remember what those models look like. The DNA is the double helix model. The RNA is the single helix model. And those break down into the nucleotides, which are their building blocks on the right-hand side. So this is what enzyme from the pancreas. And that is going to be your DNAs and your RNAs. DNAs and RNAs from the pancreas. Okay, so that's the end of those images. There aren't a lot. I want to go through my chemical digestion notes. Hold on just a moment. You should still be sharing to this page, but just in case, I'm going to break the share and come back. I'm never quite sure. Now when I do this, if I still have you with me. Okay. So here we're in metabolism. This, again, my notes and the PowerPoints give you the answers that go into all these fill in the blanks. So here we talk about these physical mechanisms of digestion. The activity type is either digests or propels. That's where that information comes from. All right. Then we get into the hormonal digestion, where we're looking at gastrin, CCK, and secretin. Then we talk about the digestive aids, bile, hydrochloric acid, bicarbonate, where they come from, what are their actions. The PhysioX, you can forget that now because we're not in the lab. doing that, but that was generally one that we would perform in the lab. And this is essentially a different way of organizing the same material, that little chart that we just filled out with the last exercise, where we're talking about, this is all about carbohydrate digestion, right? What are the different enzymes? We have amylase. The production site is either the parotid gland, from the oral cavity, or the acini cells of the pancreas, so from the mouth or from the pancreas. The target site, the substrate is starch, your end product is maltose, or disaccharide. And then we look at, okay, we could either have maltose as a disaccharide, sucrase and maltase, they're all disaccharides. What is the production site of the enzyme? This is all the brush border of the small intestine, right? The target site is the duodenum because they act on the contents as it passes through the duodenum. The substrate is maltose, and the end product is 2-glucose. Sucrase, coming from the small intestine, acts on sucrose to produce glucose and fructose. Lactase is the enzyme, acts on lactose to produce glucose and galactose. And then we look at fats. How do we break them? How do we break down the fats? Sorry, my phone was going off. Let me mute that. Okay. Then we have nucleic acids. And by the way, the lipase is produced primarily in the pancreas. Right? It's released into the small intestine, duodenum. Our substrate is triglycerides, and our end products are 1-glycerol and 3 fatty acids. All right, then we have nucleic acids. We have the DNA and RNAs. Right? They act on DNAs, acts on DNA as a substrate. RNAs and RNA, end products are nucleotides. They're formed in the pancreas. Okay, then when it comes to proteins, it's a little more complicated. I think the chart I use in the lab is easier to understand, but this is just kind of a different way to organize the same material. All right, so we have, in the stomach, I think maybe it's easier to see that the production site in the stomach, it produces pepsin to the left-hand side, right? And it acts on proteins to form dipeptides. And then we move down as this passes to the small intestine. We have trypsin and chymotrypsin produced by the pancreas. It acts on the same large proteins to break them down to dipeptides. Then from the pancreas, we have carboxypeptidase that acts on the dipeptides to break them down to amino acids. Technically, it's just leading off really the carboxyl group, and then dipeptidase from the small intestine acts on the dipeptides to break down to amino acids. All right, so I thought I might have some more pictures of molecules in here. Then I'm just going to use those pictures that we just looked at, I believe, in the image data, those images that we just looked at here. I'm just trying to look and see. I used to have lots of pictures of them, but perhaps in the future I'll add some other individual pictures. I do have some that I'm creating a separate database for all this, for all these images and these models. They'll look a little weird because they also have to be somewhat designed for visually impaired individuals. So they're going to have different kinds of labels, ovals with like a bright yellow background with letters on them, pointing to different structures. So it'll look quite different, but they're the same models and things that we looked at in AMP1 and AMP2. So hopefully I'll try to have an opportunity to load some of those up here. But evidently, thus far, I will probably be just using the images that are there on the course page. All right, so that is the end of the digestion lab. What I would say, let me not end right away, I'm sorry, is that we've looked at organs in the digestive system from last week. We looked at the liver, we looked at, the pancreas, we looked at the small intestine. So at this point, I should be able to point to the liver and let's say what digestive aid is produced by this organ. So that would be bile. Or if I pointed to the stomach, I said, what digestive aid is produced here? Hydrochloric acid. Or what digestive aid is produced by the pancreas? And I'm pointing to a pancreas. Then we're talking about bicarbonate. If I pointed to the small intestine and I ask you, what hormone, ding, 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 hormone, you only have to know 3, gastrin from the stomach, CCK, and secretin from the small intestine. Which one is stimulated by an acidic environment maybe and feeds back on the stomach to inhibit stomach secretions and to stimulate bicarbonate from the pancreas. It'll be using, that action, the actions that I gave you for those hormones somehow. Okay, so, all right, then that's it. Use the information, the anatomy that you learned in Lab 7 to help you with Lab 8 as well. All right, thank you.